All right, that was great. Thanks a lot, Dave. So welcome all. I'm really excited to see such a great turnout. And I'm really, really excited to introduce Guillaume Achaz to you. Guillaume is a colleague of mine at in Paris, whom I first met in John Wakely's lab at Harvard when we were both postdocs. And so two really nice things have happened. One is Guillaume said to me last summer, I think I'll come tour the East Coast. Would it be okay if I visited your school? And I said that would be absolutely marvelous. And then last week, John said, would it be okay if I came to Guillaume's talk? And I said, absolutely. So I've done nothing. And there's this happy coincidence that we have these speakers here. Guillaume's got a wonderful mind. He works on many things, which often means not very carefully on any of them, but this is the opposite of the situation with Guillaume. He works on many things, all of them very carefully. We spent 2 hours this afternoon in which I unpacked all the stuff that's rattling around in my head, and he was with me at every step of the way and finding connections to things he's been thinking about in his own mind. He's really extraordinary. As it turns out, he's here until midday Friday. So because I have a full day of meetings tomorrow, he and I played together today, and tomorrow he'll have meetings with folks in the community. There's a Google Sheet that Maria circulated. So if you're interested and not on that calendar, please help yourself to one of those slots. So with that out of the way, as I said, the introduction will take 5 minutes.
Thank you.
I'll give you the floor.
Thank you for the nice words. I'm very glad to be here and touring in the New England. That's very exciting, discussing with many people on different subjects. I have a talk here, but I'm happy to discuss any kind of science with any of you guys. So getting back to the talk, there is a title, so weak genetic draft. You may have heard the terms before. Let me restart the timer. Okay, so I get an idea of. So there are keywords here, and Lewontin's name appear, right, in the paradox. I have to say this is a joint work with my friend and colleague Emmanuel Scherzer. So he was appointed professor in mathematics in Vienna, so he's still there. I'll take some time to do some introduction about the questions. So probably if you're experts in population genetics, that's going to be something you already know, but if you're not, you may benefit from it, right? And we all agree on the same vocabulary. I think it's important to do that. And I chose actually the two drawings to illustrate this talk. So the first one is Don Quixote, right? As you may have noticed. So the story about Don Quixote, I think, relates to Lewontin's paradox because, you know, this is about the way we perceive reality. Right? So there is the windmills and they are moving with the wind, and this Don Quixote is completely sure that these are giants that he's supposed to attack, and you know the story, he run into the windmills and crash over it, right? So I think that's important to emphasize that there is a reality and there is our perception of the reality, and sometimes it may differ, right? And the second one is about the watches of Dali, because I think the Lewontin's paradox could be rephrased as the problem of time scale and how you perceive how time. So that's why I chose the second picture. So very generally, the mindset of much of my work, and especially this one, is the following. So the biology, here on the right, you have an evolutionary process, and here it's a simple branching process, like where you have lineages splitting and dying. Okay, that's an example of an initial process. This is meant to represent a species that is growing in size. And what you do have is observations, say individuals at present time here that are colored, the colored circles here, right? If you abstract the process, or only following the genealogy and the relationship between these individuals, you can get rid of the whole process and find out like there is a tree relating all the individuals. But the tree is mostly unknown. What you can access is these individuals there with their phenotype or nucleotide sequence. So the biology goes from from right to left. But the goal for us is to go from left to right. Oh,
Then you can
thank you,
stop
Dan.
where you are required to be and can point. Yeah.
Yes. Okay. So the game is to, how do you go from observables to the tree, but unless you're really interested in like inferring the who's closest to who, what you're aiming for is not exactly the tree, is actually the process, right? So now that being said, I'm going to do a very short historical tour of molecular evolution. It shows a few key dates. So I think one important one is 62, where Emil Zuckerkandel and Linus Pauling, they assembled sequences of proteins from different species, right? They compute the number of differences between these sequences, and they count the number of differences, and they display and they show that the number of differences, they actually correlates very well with the time divergence between the two sequences. And that's called the molecular club. So it's a divergence between species, right? In the same 60s, slightly later, Richard Lewontin and Hubby not sequence, but characterize the polymorphism of a few chosen loci within the species. And the surprise was like most loci were polymorphic. They showed diversity. Nowadays, if we were sequence the genome, we would say that most of them are, not most, but all of them are polymorphic. Right? So there is diversity, divergence between species, but also diversity within species. Based on those two observations, and among others, at the end of the 60s, it was proposed that the patterns of molecular evolution we see, polymorphism and divergence, are compatible with what we call the neutral model, right? And that was pushed especially by Moto Kimura, but also by others at the same time. Very early on, after the proposition of the neutral model, some people resisted to this idea that most variation is neutral. And Lewontin pointed out in his book, in a chapter, that actually the prediction of the neutral model, the predicted diversity of the neutral model, does not match the observed diversity in the field, okay? The range of population size does not reflect the range of diversity. And I'll go into more detail exactly on what it means, okay? And since then, and still ongoing, a very vivid debate on like, are polymorphism neutral, or are they not neutral? What's the relative importance of both? Everyone agrees that drift should exist and selection does exist, but it's like, what is the process that drives diversity is the question. What are the standard neutral models? So it's an abstraction of reality. In this purest form, you have a population, say, of constant size, and this is meant to be the population, this as a species. It has many assumptions. Among them, the population size should be constant. That's the number of individuals in your population stays the same. There is a strict pomixia. You can see it. Any individual will find its ancestor anywhere in the previous generation. There is no special structure, not any kind of structure. And there is no selection, okay? Consequence of that, the number of descendants per individual is Poisson distributed with a mean 1. All right, so that's the general one, and the specific implementation of these standard neutral models is called the Wright-Fisher model, proposed in the 20s. Here is a realization of a Wright-Fisher model. So you have a population size of, I say 7. Okay, so any column of disk is a generation, and you have generations succeeding other generations. Individuals relates to the previous generation by randomly sampling their ancestor, right, in the previous generation, and you go on like this, right? So for almost, I would say, 60 years, yeah. Mostly the focus was you condition on sampling one individual, you follow that individual, and you follow the number of descendants of that individual, right? So see here, it has 2 descendants and 3. And so what you follow is the frequency of the descendant of that individual in the whole population, okay? So that's I'm going to refer it as genetic drift. That's the forward view of the model. And since the 80s, there is another way of looking at the same model, is same realization, is looking at it backward. And in this example, I've sampled. So this, by the way, this is a real realization of the Wright Fisher. So I've sampled all the individuals here, and I will follow the ancestor back in time. So see, there is an ancestry, and there are events here that go coalescent event, and there is a tree emerging from that model that is called the coalescent tree. So the coalescent tree is simply the backward version of the forward process. Sorry if it's very elementary, but I think it's important. Okay, so the paradigms in which we do our studies is the following. We have individuals. As a good geneticist, we couldn't care less about the individuals, so we reduce them to the genetic material. And we measure diversity as we are interested in population. So diversity is increased by mutation that occur at every generation, right? And it is removed by genetic drift that you can see either forward or backward. Importantly, the time scale at which it will remove diversity is capital N, being the population size. So at equilibrium, you add mu per lineage per generation, you remove diversity. So you have an equilibrium value of n mu, whatever is the exact measure you will find. This is the equilibrium value between input and output of diversity. I'll say a word on the molecular clock. So there are many ways to see the molecular clocks, but I like this very pictorial way. So you may have noticed that if you follow the descendant of any given generation, so you follow the descendant of every individual, all the descendants will go extinct except for a single individual, right? And that single individual will have one descendant that at the same time have the same property of being a founder of the population in the future. So there is a single line that will connect all these individuals that will be the founder of the population in the future. So what is the molecular clock? One way of seeing it is simply the mutation that fall in this lineage, right? All the other ones, they will be gone in the future. So you have a constant rate that is independent of population size that will tick and put mutation on that lineage, right? And from this point, we don't know who's going to fix or not. So there will be the coalescent tree at the end of the process, just because you stop the process here, right? So that's a way to see the molecular clock that will be useful for the rest of the talk. Okay. The last important thing, I think, is where is this idea of the neutral theory what was the motivation to propose the neutral theory? I think the first argument is to say that there is a cost to natural selection. So it was proposed by Haldane at the end of the 50s, and he had in mind the moth that were either white or black, and you know all the story about this moth. So to select for a color of the moth, that means you need to have at birth more individuals. And the selection is actually the killing of some of these individuals. So to handle some selection, that means at birth, you need a lot of individuals, right? So if you can compute and you can crunch the numbers and compute how many individuals I need to have extra at birth to see the selection operating. So there is a cost for any trait you're going to select. There is a cost of selection to this. So that in the end, you cannot select for an infinite number of traits at the same time, otherwise the cost will be too high, okay? So the calculation of Haldane show that should be on the order of hundreds. Other models without calculation end up with being thousands or tens of thousands. But still, the idea is there is a limited number of variants you can select at the same time, okay? You can't select every single polymorphism. The cost will be too high. So because there were many, many polymorphisms, that means most of them would be neutral, right? So that's the first point. Second point is the divergence of molecular clock occurs at a constant rate, right? So there was lots of discussion about this molecular clock argument. Should it be constant per generation or per year? Why does it vary from locus to locus? If we count the amino acid replacement or the neutral or the non-synonymous on the synonymous mutation, should it be different or not? I mean, there was But this is the second argument that was right at the beginning on the early papers. Slightly later on, few years later on, the third argument was put on the table that diversity, the observed diversity, exceed the mutation positive selection equilibrium. Usually mutation selection is mutation negative selection, and here, so positive selection will erase diversity at the scale of 1/s generation, right? So if you want to have a non-zero diversity, that means the mutation should be keep putting more and more and more variation at each generation, right? Whereas at the neutral scale, they remove diversity at the scale of capital N generation, which is much slower. So you can account for much smaller mutation rate. So that was the third main argument. Okay, that being said, the question is the following. Okay, we have a null model that is proposed and well established and used by many of us. That is the fundamental basics of any super sophisticated software you'd be using, like the last PSMC version, following the trajectory of these bugs you're interested in. The question is like, is this the right null model, right? Is this the best null model we can have given what we know, okay? And to exemplify this, I will use that example. So what we do have on the left here is the model population. So exactly the one I described. So it has a capital N number of individuals, constant population size, strict parmixia, no selection, put more into the wish list if you want. Under this model, if you sample 2 individuals, as I said, their common ancestor lived around capital N generation in the past, okay? Given that, the diversity, the number of differences between 2 individuals should be 2 times this, so going backward and forward in the tree, times the mutation rate. That's the prediction of the Wright-Fisher kind of model, right? That's the model world. And on the right side, you have the real world. Anything you've put on your list here will be wrong, right? Constant population size, no way. Strict parmixia, no way. Everything is structured, right? No selection, we all know there are plenty of selection ongoing. So in this realistic population, that probably completely differ from the model population, the population geneticist, and I would include myself into that, invented the most beautiful construction. It was called effective population size, right? So effective population size, I would argue, is the way to fit that real population into this completely simple abstract model, right? So you want to squeeze in your real population into your Wright-Fisher kind of model, but for that, you have to have a time scale, right? So that time scale, even in a real population, if you sample 2 genomes, haploid genomes, there will be a common ancestor in the past. That common ancestor, so the horizon to the common ancestor, let's call it effective population size, okay? So effective population size is the number of generations you have to go back in the past in a real population to find a common ancestor. Obviously, in most of the cases, you don't have access to this effective population size. But what you could do is leverage the diversity, right? Divided by the mutation rate and gets an estimate of the effective population size. So the effective population size, in that way, is an estimate of the common ancestor, of the time to the common ancestor in a real population. All right? Now, if you look at that, this is the model, and capital N, the number of individuals, is the expected timescale, right? If you accept this model as reality, it should be capital N. And NE is not the expected one, is the observed timescale. When actually did the ancestor lived? Was it capital N generations ago? Probably not. So let's call it NE, effective population size. So that's one way of defining effective population size. So once you've done that exercise, you can compute numbers. And I put the HIV in first because that's exactly when I started my postdoc with John. We are working with the HIV. So within a patient, there is about 10 to the 10 number of viruses, right? That's the count. The biology says that. If you compute effective population size, you end up with 1,000. So either what I've done, you accept that using your favorite software, you're not going to put 10 billion, but 1,000, and you put 1,000 in your software and you do your prediction, or you ask yourself, well, am I ready to accept a factor of 10 to the 7? Or... But that's, you could argue this is only HIV. Well, let's take humans. 10 to the 10, effective population size, 10 to the 4, factor of 1 million. E. coli, number of individuals, I asked my colleague Olivier Tonin, we say, well, about 10 to the 20, maybe 10 to the 21 worldwide, right? Effective population size, the number of generations have to go back in the past, 10 to the 8. Here we're talking about a factor of 10 to the 12 or maybe 10 to the 13. And you can go on the list. This one is especially fun. That's a crinoid that lives all around the Antarctica. And if you estimate the effective population size, you get 10. It's as if the whole population were 10 individuals. Compute number, Drosophila, C. elegans, whatever species you like, you always find the same answer. This effective population size is always way, way smaller, orders of magnitudes lower than the census size. Question becomes like, why diversity does not scale with population size? And that's the Lewontin's paradox. Okay. So in the original framing of the paradox, it was just to say that the census size have a huge variation, but the diversity has a very narrow variation. I'm willing to take a step further in that direction. Is like not only they don't relate, but the effective population diversity is always way under what standard neutral model would predict, right? Way under, orders of magnitude, up to 10 to the 30. This is a nice plot with the similar ideas. They collected many, many different species from different clades, measure diversity, and see the range of diversity is maybe on 2 orders of magnitude, right? Whilst the census size will cover, I don't know, 10 to the 20 orders of magnitude. So there is a complete disconnection between like the population size and diversity. Huge range for hand, very narrow range for hand. At that point, you're confronted to yourself and say, okay, either I'm going to accept to put those magic numbers NE into the softwares and just do the empirical predictions. And it kind of works somehow for many cases, just using the magic number, give the right prediction, which is amazing. Or you start like asking yourself, why are those numbers so different? Right? What caused this? So you follow the white rabbit into the hole, like asking the question, why is diversity or time, coalescent time or NE or whatever you phrase it, is so small compared to what we expect in the model? Next step is like, okay, but what's this effective population size? So this is one definition of effective population size. If you go through the literature, you'll find dozens of those, and they not always give the same answer. So you start asking yourself, yeah, but what is this effective population size concept that we invented 100 years ago? And then even worse, you start asking yourself, yeah, but what is genetic drift? I mean, they told me this is about sampling at every generation. Do we really sample stochastically like this or not? And then you end up like, okay, you should change your target. I mean, it's completely insane. We are tweaking numbers for some magical reason. We put magic number, it works. Why? What is going on, right? What is going on? So that's a way to rephrase the Lewontin paradox. So I reached the bottom of the well. I was like really asking myself, should I go on or just stop doing population genetics at this point? Eventually I recovered. So the one of the output of this is really examining what is effective population size. And there are many, many, many questions you can list. What are those magic numbers? How do you define them in the model? How do you measure them, which is slightly different? Definition, metrics. How do you infer them in the real world, right? With real data. What do they mean in models, in data, et cetera, et cetera? How are they used in conservation genetics? How are they used in population genetics or in adaptation? And they have a different definitions, different way of inferring, different usage. What really helped me is walking through the history of NE. So we've been rereading, with the help of my colleague, Jean-Baptiste Grotevol, who is a historian of science, rereading everything from scratch, like refining the definition, the early definitions, and how it has been used throughout the history. And the fun part is access to the mailing between the scientists. So we had like letters of Sewell Wright saying like, okay, I think I don't understand what is effective, which is quite reassuring, right? Okay, so this is not what I'm going to talk today. What I would like to discuss about is Lewontin's paradox. There is a recent, I would say, generous review by Brian Charlesworth and Jeff Jensen last year. So the first idea that comes to your mind is like, okay, this idea of whole pomixia is probably wrong. Everything is structured. Locally, if you sample two individuals locally, because they are structured, that means they are more inbred somehow. So their common ancestor should live closer in the past than if they were pomictic. That's absolutely true. But if you do sample individuals from different areas, you have the opposite effect. So it tends to actually delay the common ancestor. So structure, even though locally it will reduce diversity, it globally tends to inflate diversity. So I don't think this is a good way to solve the paradox. The typical answer, if you work in human population genetics, they will serve you the demographical scenario, right? Say, population size is 10 to the 4 because probably humans were 10 to the 4 in the past. Are we ready to accept this explanation for every single species on Earth? Where we really 10 to the 4, I mean, if the gorilla, they're almost extinct, the 10 to the 5 or 10 to the 6. 10 to the 4 is a very small number for a biological species, right? And plus, to have an E of 10 to the 4, that means in the last 10 to the 4 generations, the population size should reach 10 to the 4 individuals, right? So for E. coli, if you go back 10 to the 8 generation in the past, the population size should be below 10 to the 8, which are extremely small numbers. That's a possibility. Leave you think about it more. It's possible. Another explanation I found particularly interesting is it was proposed by a colleague. He said, you know, these numbers, these population, these timescales of drift doesn't mean anything, right? If the population gets very, very, very large, it then fragmentates it to new species. So anything that we call species is something that has a founder effect in a recent evolutionary timescale. Okay, that's another possibility. And finally, the classical one is selection through linkage. So here, the idea is selection will not affect every single polymorphism, but only a single locus selected will have an effect, not only at that locus, but on the rest of the genome as well. So that's what we call linked selection. There are two types of those. First one has been named background selection. That's the effect of deleterious mutations. When you have deleterious mutations occurring in a genome, because that genome will not invade the population in the future, it will reduce the total number of individuals that will contribute at each generation, right? And the other one is positive selection, and this has been called a genetic draft. I will be focusing on the second one today. I have an argument to think that background selection is not the solution to the problem. I'll come back to that at the very end of the talk. Okay, I think now we're ready for the genetic draft. Sorry for this long introduction, but I think it's important too. put the question in the right, well, in one angle, I can have some contribution, right? So the weak genetic drive. So actually, what I will be pushing for is not only the genetic draft, but a regime that was underappreciated before that we named the weak genetic draft. But another liar, before talking about genetic draft, you had to talk about hitchhiking. So hitchhiking, it's an explanation of linked selection that was proposed to solve the paradox in the middle of the 70s. There will be some reference above, and don't gonna take the time to go all through the reference, and it's very generous, bibliography. So that's the forward version. So now your genome is a two-loci genome. Okay, you reduce your genome to the simplest two-loci genome. You have one locus that is selected, and that's going to be the red square. And you have a neutral locus. Okay, so any polymorphism that fall into the neutral locus will have no impact on fitness. Everything is important for fitness will be at the selected locus. And there's partial genetic linkage between both loci. So here I have a right Fisher population of 5 individuals composed of these two loci. Okay. At some point, a beneficial mutation appears in the selected locus, and as any good beneficial mutation, it would invade fairly quickly the population. So after a few generations, everyone has the beneficial mutation. Okay. What we are interested in the hitchhiking model is not really the fate of that red allele, right? Is the fate of this gray allele that happens to be in the same genetic background than the first beneficial mutation. So because these two are linked, physically linked, it will increase in frequency, and it will increase up to the point where it will invade the population. But wait, see, sometime there will be recombination disconnecting these two individuals and reconnecting, say, some allele from the white population to this selected locus. So along the way, it will increase in frequency, but it will not reach 100% unless you have no recombination. The effect is tuned by the ratio between the recombination rate and the selection coefficient. Like, say, if A is very small, say recombination rate is 0, the effect is very strong, right? Recombination rate gets high, not so much an effect. Selection coefficient, the higher it is, the stronger is the effect. Okay, that's the forward view. And 10 years later, a backward view of the same process, again, forward, backward, was proposed. So here is this selective sweep. You're following the frequency of the selected allele. If you look at the genealogy of the selected locus, it will all boil down at worst in this individual. That's the first individual that originated the mutation, provided that the mutation occurred only once. That's for German. It's working on recurrent mutations now. A sweep, as I said, lasts for about 1/s generation. Because lineages do not recombine, they will all crash during this sweep, right? At distance 0, all lineages coalesce during the sweep. If you allow for recombination, these lineages, they can escape through recombination. So the model becomes like you're following the genealogy of the neutral locus. It's constrained by the sweep here, but eventually it can escape this. So it becomes a model of coalescent with two compartments. One is invading population, invading allele, and the other one is the resident, right? And you can move in and out using recombination. Because the sweep lasts 1/s generation, each lineage recombine at a rate of C per generation. So that ratio C/S, you can see it as a rate of recombination per sweep and not a recombination per generation. That's a way to have an intuition about C/S, right? So if your rate of recombination per sweep is very high, then you're going to escape it easily. If not, you're not going to escape it. So pictures, if you sample individuals at the selected locus, they will all coalesce for the last sweep. If you sample individuals at a neutral locus, some of the lineage will be trapped by the sweep, others will escape and enter back the very large populations here. So the original derivation of the hitchhiking was done on an infinite population. So later on, there was attempt to do some finite population, and we thought it would be worth re-examining this finite population. So in a finite population, if you have a right Fisher plus selection, so you're only following the selected allele, right? It will go up and down because there is some stochasticity, but it will tend to invade, right? But sometimes it's not going to invade, it appears and disappears, right? So that's the stochastic process, starting with 1/n individuals invading the population or to a frequency of 1. Turns out that an excellent approximation of this process is the following, that we name the Riff model, where you draw randomly a random starting point. And that's not 1/n, that's an exponential random variable of mean 1/2s. You do a deterministic, like infinite population trajectory, and you stop it at a random final point. It's also 1/2s, right? So this turns out to be an excellent approximation of what's going on with the stochastic model. So it's a semi-deterministic in a way that only 2 random variables are drawn, one at the beginning, one at the end. So you can already picture that simulation-wise, it's much, much more efficient than this, right? So we named it the Riff model, talked to my friend Amaury and said, hey, you know, I've derived this same model, but 10 years ago. That's how it is, 20 years ago. So we had to acknowledge that this semi-deterministic model was already there. You can compute the average time to the sweep. And as I said, it's mostly 1/s plus some numbers, times some numbers. But they didn't derive at all the coalescent under this model. So we derived the probability of coalescent under this approximation. I'm going to skip the math for today, but here are the results. And it looks like this. This is the probability of the coalescent for a single sweep. Okay. It has a component of a contribution of the population size that is here. And there is all that thing. So I don't expect you to understand everything and why it's like this, but this relates to the probability that to coalesce the two alleles, they must be inside the sweeping allele, right? So because they can move up and down, you have to condition that at some point they are in the same compartment to coalesce. That's this contribution. And given that, they could coalesce during the sweep, because in the Riff approximation, you go from a random variable to 0 with a single generation. You can coalesce because you're at the onset of the sweep in the approximation. I was quite happy with that, but Emmanuel wanted to have a more elegant formula and especially not numerically integrating these things. So he worked a lot, and I have no time to discuss, but he used a much more elaborate diffusion approximation and came out with another formula, which is this one, has the same shape, right? Looks. The only thing is that these two integrals now are the simpler thing, okay? So both approximation have the, I would say, the canonical form of the usual approximation of coalescent under sweep. There is a contribution of population of NS, and there is a constant that is either this or that, depending on the approximation, okay? So how good are these new approximations? So what we did is simulate a forward, right Fisher, or backward using the Riff model, right? And the simulations are the dots here, right? So circles are the backward ones and the forward are the triangles. As you can see, well, they kind of both agree, which was the good news. The theoretical predictions, the previous approximations are here. So, and I'm evaluating the constant now. Right? So the previous approximations are here, whereas the new ones are here. So it seems to be much better than the previous one. That's for when you vary the census size, when you vary the ratio C/S, see, when the ratio is very small, say smaller than 0.1, most of previous approximation and new one, they give the right answer. Interestingly, if the ratio gets larger, say 0.3 or 0.4, our new approximation, they fit quite well the simulation, but all the other ones, they completely underestimate that constant, right? By kind of a lot, right? Importantly, these new approximation and also the previous one, they explode at 0.5. So if you look at how they behave around 0.5, they go to infinity. So they're only valid for a certain parameter space. Okay, so we have new approximations that are good for large n, and for a, say, smaller than 0.45, otherwise it's tend to ours overestimate and the previous underestimate what's going on in the simulations. All right, so now we're ready for the genetic draft, recurrent hitchhiking. So that was the term genetic draft. At the same time, it's a play on word with genetic drift. But I think I asked a native speaker, they also told me that draft is when you have a first biker, all the other ones that are behind the first one get aspirated by the first one. I did not realize this since 6 months ago, but that's how it is, right? So it also has the same idea that hitchhiking, right? But now, instead of having a single event, you have many recurrent selective sweeps ongoing, okay? So if you look at the selected site, again, they all collapse at the first last selected event in the past. If you look at the neutral locus, and let's allow yourself to have this neutral locus far away from the selected site, okay? If it's far away from the selected site, this ratio A, C over S, is quite large. That means for most of the sweep, the lineages, they don't even feel the sweep, right? So there are many sweeps occurring, but sometimes one of the sweeps will coalesce two of the lineages, right? Here there is a coalescent event. And notice that the coalescent event here, they have nothing to do with genetic drift, right? They are only driven by the rate of selective sweep. So can it work? Yeah, you know, let's do some simulation and see how it goes, right? So this is a real simulation. These are the sweeps. I was monitoring the recurring sweeps, and I recorded the last. So I sampled the whole 10,000 individuals, recorded the whole tree, and looked where happened the last coalescent event. And indeed, they all coincide. They all coincide with selective sweeps. So really, what's driven the tree are the sweeps, right? And C, they are far away from the selected locus, so that in many cases they don't feel the sweep, right? Right. So we were there, and here in that example, the time scale, the average time scale was 3,000 instead of 10,000 for the tree. Pardon me?
A is 1/20.
Yes, in that case. Yes, something like this. We had another problem, like increasing the population size will increase the rate of sweep because it depends on if the mutation rate is dependent from the population size, you have more and more sweeps as you increase your population size. To prevent that, because we were interested in deriving mathematical things, we came up with a trick, but that's only a trick, that environment will change regularly at an exponential rate of lambda. Anytime the environment changes, it triggers a new selective sweep, right? So that prevents the selective sweep to be nested one until the others. So we are currently working on like nested selective sweep, but that's a trick to separate the time scale, right? And now you can characterize the model pretty easily. You sample two lineages at the neutral locus, and anytime you have a sweep, you toss a coin, does it coalesce or did not, right? Because you can compute the probability of coalescence that becomes a geometric with this probability of coalescence. So environmental changes that are not necessary, but that's just a trick to say you have different sweeps, and that's going to tune the coalescent rate. Okay. Now, output of the model. First of all, and I think this is a very important argument, the diversity in this model does not scale with population size anymore. So it turns out that the diversity scale as the power law of the population size. Remember, ns to the power of 2a, right? A being smaller than 0.5, say. So that means that the diversity, expected diversity, will not depend directly on population size. You break the linearity between population size and diversity. If you're very, very close to the sweep, say 0.01, so very close to the sweep, the effect of population size almost completely disappears. So sorry, I have to say, this is a log-log plot. These are the simulations forward and backward, and you can notice that the backward are much more fast, much more efficient, so you can explore a larger population size. This is the drift, what you expect if it were drift, and the dotted lines is the analytical predictions, okay? So if you're very, very close to the sweep, diversity becomes independent of that. And this is what we call now the strong draft, and has been explored by among many Koop and Raff in the last 10 years. If you allow yourself to be a little bit further away from the sweep, say 0.1, see the diversity scales as a power law of the population size, but the slope here is still pretty flat. So that's the regular draft, but in a weak draft regime, you're even further away from the selected site. And here, see, diversity scale as square root of the population size. So now in this model, the predicted diversity is not anymore linear with population size. It's if you accept 0.25, it's going to be square root of the population size, which means a much narrower range than the population size. And if you increase up to more than 0.5, you recover the drift, right? All right, so timescale is different. So what data says, I've been collecting data and see if what's the relationship between diversity and census size, and that's a gift from my colleague Benoît Dabols at Montpellier. So this is not the population size, this is the range in kilometers squared, right? The proxy for population size and diversity of the island birds. If you put them on a log-log plot, it looks like a line, right? With the eyes of the face, at least. Pinniped, someone pointed me that the pinniped have the same property. And you know what? Last year, someone published a big data set with many diversity estimates of population size, and he claimed that actually the best relationship is a log-log linear thing, which is a power law as well, right? So probably there is something in the power law. Getting away from linearity between diversity and population size is a good way out. Second model output, and I'm almost done in maybe 5 minutes. Don't panic. Yeah? Is the so-called site frequency spectrum. So that's the frequency spectrum is simply you scan the genomes, collect all the possible variants, collect their frequency, and compute the distribution of these frequencies, right? So here, if you do that for the Yorubas, 80 million variants, this is public data. If you normalize the SFS to 1, you get something like this, with many polymorphisms that are only carried by a single haploid genomes, and not so many by this. But looking at the picture, you don't see anything really, well, except for the few first bins. So we've been advocating for a transformation of the SFS by dividing the observed value by what you expect under the standard neutral model. So under the H0, it's flat. And now you can see that there is an excess of low frequency variant that you saw here. But more importantly, there is a huge excess of high frequency variant there. And we call that the U-shape SFS. So what does a model do? If you compute the SFS as the selected locus, that's the red line. So it's L-shaped. It doesn't have any high frequency variant. If you allow A to be, say, between 0 and 0.1, you get this nice U-shaped curve that has been described. This is a strong draft regime, but let's get further away from the selected site, 0.1, 0.2, 0.3. So say, see, 0.1, 0.2, 0.3, we are almost there. The site frequency spectrum, it looks almost neutral. But remember, if it's 0.25, the time scale is square root of the population size, right? So it's as if these models, they produce neutral patterns that if you apply your favorite TouchGMSD, you won't see anything. Probably you won't see anything, but still the time scale is completely reduced, right? So that's an interesting one. That's the picture at the radiation, very close to the selected locus. You have multiple merger coalescent arising that are typical of U-shaped side frequency spectrum. But if you get further away, if you're not so far from the, well, you're far away from the selected site, but not so far, time scale is distorted. Still, it looks neutral, right? Interesting. What about the real genomes? Are they showing like a zest of U-shape? So we've been analyzing 45 species from all around the tree of life, like Bacterias plants, characterize the U-shape. They all show a U-shape kind of a SFS, more or less, with more or less strong U-shapedness, right? But so if you analyze genomic SFS, regardless of the species, you tend to see those U-shaped things, right? By the way, humans, they're here. They're not there. Okay. Last but not least, the molecular clock. I told you that the molecular clock stems from the fact that you have a single lineage persisting through time. That's true for the standard neutral model. If you increase this model with demography, you have the same property, right? So the molecular clock is not distorted by the variation in population size. If you add structure, well, you have the same property again. So molecular clock is not distorted by structure. Interestingly, with recurrent positive selection, sometimes you have like a mutation, say the red one that will invade fairly quickly. And later on, a blue one that will invade and a green one. But except for these rare mutations that are the positively selected ones, all the neutral mutations that will fall in this line will follow the molecular clock. So provided that the number of positively selected mutations is very small compared to the number of neutral mutations, you have almost no distortion of the molecular clock. Okay, time to wrap up. The question is not, is this model a real model? I'm not so naive to think that life is like not the Wright Fisher, but the simple genetic graph. The question is like, is it a better null model? I would argue yes. So the visual on this is the following. You have sometimes, you don't need a lot, positively selected events occurring, right? That will wipe out more or less efficiently diversity at the selected locus. In the near vicinity of the selected locus, you have a reduction in diversity and a tendency to have a U-shaped, a multiple merger U-shaped side frequency spectrum. But more importantly, maybe 10 times larger on a larger zone, you erase diversity, but you have a neutral tree, you have a neutral pattern. So applying any neutrality test in these large regions of weak draft will not show any deviation from the neutral model, except that the timescale will be reduced, right? Okay, in words. We all agree that sequence do change through time. That's no question. We all agree that homologous loci show diversity. That's no question. The question is, what is the cause of the molecular evolution? What tunes the diversity? What is the proper process that remove diversity because we're having not enough diversity, right? So neutral theory proposes that diversity scales with population size. Importantly, NE is the tool to put your real population into the model so that actually NE is inferred using the diversity. So you have a nice example of tautological reasoning here, right? Positive link selection, it's a potential good candidate to help you getting out of this. The first thing is breaks the linearity with population. So I think I would advocate maybe not for this model, but for any kind of model where you don't have any more the linear relationship between N and NE. Problem with background selection is you don't break this linearity. It's only a fraction of n that will not participate to the next generation. So you're going to put a constant, say 0.001, but 0.001 and 10 to the 21 is still 10 to the 19, right? So I don't think that's going to help you fixing the Lewontin paradox. Positive selection does not need to be very frequent. It just needs to be frequent enough at the scale of drift, which is capital H generation, which is a huge time, right? So just need to have positive selection now and then at the scale of the drift, right? And it will dominate, the process will dominate over drift. And there is no need for a caricatural sweep. I mean, my colleague Emmanuel was so willing into doing the math that we had to do a caricature, but there is no need to do that. All right, I'm done. Thank you. I'm happy to ask any questions. Please.
Thank you for the really exciting talk. So I have two questions. The first revolves around, I guess, something that I don't think I've read on this topic, but do we know anything about the relationship between like fluidity in these ranges between, you know, diversity and the expected like population size? And then like,
shall we first answer this first question?
Yeah.
Okay. You mean beside the general comment that microbes tend to be haploids and have much larger population size than diploids, or?
I'm thinking more about like plants where you think they like have multiple fluidity levels for like different reasons, whether it's like you know, you just double your fluidity, or you can have essentially two diploid genomes stacked together.
Except for the wheat, for example, and all these plants that have domesticated us so they can have a giant population size. I don't know the answer. I don't know.
And my second question is, I am on board with most of the demography arguments that you were talking about, but like one demographic, I guess, event that is much more appreciated now than historically is gene flow. And I'd be willing to bet that, you know, gene flow has kind of been constant and there's never been these really rigid species barriers, and that it's been leaky all throughout evolutionary history. And I guess my question is, how does, what do you think your model would say in the face of gene flow?
Finish your question.
Oh, just again, the interplay of gene flow, and then essentially like potentially even more sweeps, but now also maybe some more like of the negative link selection as well.
So, as I mentioned, well, gene flow relates to the idea that there is structure, right? So if you do have structure, you tend to have an inflation of diversity because you retard, you put further in the past the ancestry between for the neutral ones. Now, whether the selected regions appear by mutation or by introgression, really that doesn't make so much difference in the model, right? So you have, what you need to have is positively selected events, regardless of their origin, could it be introgression or mutation. That's more or less okay. Just what's going to do is wipe out diversity around the targeted region more or less efficiently. Remember that the sweep is 1/s, so 1/s is not 2 generations. So you still have time actually to regenerate. Even during the sweep, you will generate diversity. But importantly, it will also wipe out diversity on a very large region, right? Again and again, most of the regions will not fill the first event, but event after event, it will like reduce diversity on a very large region. So I think this model doesn't say anything about introgression. If the introgression is neutral, then you come back to the problem of structure, and if it's positively selected, it's the same by the mutation. Most welcome. Thank you for the question.
Can you give some intuition for why the number of chromosomes of the organism doesn't enter into your model?
You mean the ploidy or the number of chromosomes for the hap?
So the number of chromosomes is, you know, sort of roughly the number of linkage units. So among the organisms you considered this quite a bit, you know, at least one order of magnitude difference. Why is that not important?
Well, I think it is important because if you have two loci sitting on two different chromosomes, their probability of recombination is 1/2, right?
Right.
So if you have a single event at the same time, only the chromosome hosting the beneficial mutations
Right.
will be affected. But now, change your view on the world. Say there are hundreds of selecting events occurring at the same time. A typical genome has like 10 or 20 chromosomes, right? So it's, if you allow yourself to have hundreds of selective sweeps ongoing, it's very likely that all the chromosomes will be affected. Though, the size of the chromosome matters because the recombination rate relates to the size of the chromosome. So I haven't tested that. That's a good possibility. Is the chromosome size important or not? Well, there will be a balance because if you have a large chromosome, you have a higher chance to have more than one sweep ongoing. But at the same time, the recombination map will be different. So I don't know. What I can tell is many people have looked at regions of different recombination rate. And that was also one original support for the selected mode is like regions where recombination rate is very low tend to be lower in diversity. That was proposed as a good support for the effect of link selection. So that's what I can tell about this. Does it satisfy your curiosity?
Somewhat, yeah.
Okay, thank you. Yes, please.
Yeah, I also had a question. I think it's probably related to the recombination rate and some size issue. Yeah, I was thinking about like how the distance from the selective site would scale with at lower recombination rates, with like an extreme example of microbes having often no recombination.
May I challenge the view of no recombination in the microbes? I think microbes do have a lot of recombination. It's just that the recombination machinery is slightly different. So you replace an entire section of the genome instead of having crossovers. So they don't have the crossovers, but they certainly are extremely mosaic to the point where people think that we don't have a tree for the microbes. It's a network, right? So there is all these people with very solid arguments saying like there is so much horizontal transfer, and that's the one you can detect. But when it's within population, I mean, there are plenty of signs of recombination. So as far as the effect of recombination, as I said, low recombination regions tend to be poorer in diversity. So that's probably, yeah. Well, that doesn't validate the model, right? But it's coherent with the model prediction. Yes, please.
How about dominance? And I guess that relates to Dave's question about Floyd. It's the same question. Do you just assume that there's a random value for dominance across all these mutations, so that it doesn't matter? Certainly regions that are haploid versus diploid would have very different dynamics for various models of dominance versus recessive. Whether that's a noise factor in your models or whether that's a predictive thing. 25 years ago, Dan and I had a project that empirical sequencing results came out exactly opposite what we predicted.
That's life. That's always like this, right? You put a prediction and validate, and real life is being tough with you. That's expected. I don't. In a caricatural sweep, say you have a beneficial arising, increasing frequency up to the point of fixation. Really, dominance doesn't. I mean, it's going to change slightly the time scale of it, but still it's going to be 1/s. So the rough time scale of a sweep, regardless of its recessive or dominant or codominant, the same. But if it's over-dominant, things change completely, right? Because you have balancing selection arising. So I would say, if you assume dominance is between recessive and dominant, that's not going to play a major role. But if you allow for lots of over-dominance and lots of balancing selection, that's going to change the thing. But in the wrong way, that means if you have many balancing selection, it will inflate diversity. And the question is why diversity is too low. So I don't think over dominance will solve your paradox. That doesn't mean it doesn't exist, but
Part of the genome, chunks of the genome were overdominant, other chunks are.
yeah, it's probably what happened. The question is like, what's the fraction of the genome that is over dominant? So these inversions with like selected mutation, they tend to have a show like over the dominance pattern. Do they represent 0.001% or represent 10%? I don't know the answer. I don't.
Thanks.
Yes, please.
One more question.
Yes.
So suppose you have something like this inversion polymorphism on chromosome 17 that is favorable in one sense because it favors fecundity. So you can't really have a sweep in that situation because it just takes many, many generations. So how do you model that kind of favorable?
What you have in mind is an inversion
Okay,
that is
so
positively
I just-
selected, or can you
No,
be more
okay, so
specific?
I just gave an example.
Yeah, yes,
of
please.
Why this is real. So what I have in mind is a mutation that is favorable because it favors having more offspring. So there is such an example in humans of an inversion polymorphism on chromosome 17, and both alleles survive in the population because one allele favors more children, but is phenotypically unfavorable for the person who carries it. That, that's interesting for other reasons, but my question is, suppose there weren't this balancing factor, you just have an allele that achieves its benefit by favoring more offspring. So you can't, I don't see how you have a sweep in that case, it just gradually.
So you mean an allele occurring within the inversion?
Just in general, an allele that is favorable phenotypically, not directly for the individual who carries it, but because the benefit is more offspring.
Wow. To my simple mind, having more offspring is exactly a positively selected allele.
Right,
So-
but it takes many generations to have a sweep.
Wow, sweeps do take time, right? They do time. They take time. They take 1/s generation on average. So, and I mean, when we have the caricature drawing of a sweep being like a single point in time, it's because we put it in a drift timescale. Then they become a single point, right? If you forget about drift timescale, but count the number of generations, say an advantage of 1%, it's going to take you, say, a few hundred generations to fix. So a few hundred generations is not 2 generations, right? So I don't see why the inverted the inversion, if that's the favorable allele, will be different from another one, except that it's not going to recombine within the inversion.
Right,
So-
I mean, so the theory is that the inversion persists because there's this that's favorable in one respect.
Then you're in the balancing selection thing, and I completely agree. If you're in the balancing, you have no sweep, but again, that predicts an inflation of diversity, and this is not what we observed on average. So that doesn't mean it doesn't exist. I mean, there are clearly cases of balancing selection. Not challenging that.
Okay. Thank you all. Thank you.
Thank
Guillaume.
you. Thank you. Thank you for the questions.